Hi everyone, it's Dr. LeBlanc with Unit 14C on Transduction and Transformation. Our learning goals are to understand specialized and general transduction using viruses, and to understand transformation using plasmids, and to understand the concept of horizontal gene transfer. Transduction is the process of having genes transferred from one organism to another by a virus particle. The transfer of DNA from one bacterium to another via bacteriophage can be either generalized transduction or specialized transduction. What you're seeing right here is generalized transduction, when the host chromosome is broken up into small pieces and one of those pieces gets incorrectly packaged into a virus capsid and then transferred to a new recipient cell. Specialized transduction only occurs when a virus genome has integrated into a bacterial genome and picks up the genes that are next to the site of integration. When the virus DNA is excised, it picks up a piece of the adjacent chromosome DNA by accident. Here's a diagram showing just that. A virus will inject its DNA into the host cell. It will integrate into the host chromosome, right here, and then when it's induced to excise, when it comes out of the bacterial genome, it picks up a little piece of the bacterial chromosome with it. This is specialized transduction. Let's look more closely at generalized transduction. Here we have a phage injecting its own DNA into a bacterial cell that is His plus and Lys plus. The host DNA gets hydrolyzed into small pieces, and phage DNA and proteins are made. The new phage assemble. Occasionally, a phage encapsulates a piece of the host cell chromosome by mistake, right here. The rest of these phage particles have phage DNA inside. The new phage are released when the bacteria cell lyses. There are transducing phage along with the regular virus particles. The transducing phage then injects its DNA into a new recipient cell. In this case, the new recipient cell is His minus and Lys minus, and the DNA being injected is His plus. Via homologous recombination, this His plus DNA can replace the His minus DNA of the recipient. The transduced DNA is recombined in the chromosome of the recipient cell, and now the cell becomes a recombinant bacterium with a His plus and a Lys, the original Lys minus gene. It's transformed its genotype from His minus, Lys minus to His plus, Lys minus. Transformation occurs when a bacterial cell takes up DNA directly from the environment. Bacteria cells die all the time in a large population of bacteria in an environment. When the bacteria cell dies, it releases its DNA into the environment, and some of that DNA can be taken up directly by a recipient cell. So here we have a cell that is Lys minus, and it has found some DNA that is in the environment. That DNA happens to be Lys plus. The first thing that happens is that the DNA fragment binds to a cell surface receptor of a competent bacterium. Then an extracellular endonuclease cuts the DNA into smaller fragments. So it's an endonuclease that's on the outside of the cell, cuts the DNA into small enough pieces. One strand is degraded and a single strand is transported into the cell via an uptake system that has a lot of complicated proteins associated with it. After the DNA strand is incorporated or brought into the cell, it aligns itself with the homologous region on the bacterial chromosome. Okay? Our DNA from the environment is a lyse plus gene, and our recipient host cell is lyse minus. A heteroduplex forms, and via homologous recombination, the new DNA is taken up into the chromosome of the recipient cell. And now we have a transformed bacteria that used to be lyse minus, and it's now lyse plus. Transformation in the lab by molecular biologists is a little different. We usually start out with plasmid DNA that's been highly engineered. It has an antibiotic resistance gene for ampicillin resistance or for tetracycline resistance, for example. And it has our cloned piece of DNA that we've inserted into the bacterial plasmid. The first step is to make cells competent by capturing them in log phase and immobilizing the adhesion zones, which are small holes in the membrane, with cold temperatures. Log phase cells are rapidly growing bacteria that often make mistakes when they're making their cell membranes and cell walls. They have little holes in them. Not a lot in the population, but enough that we can make it useful for molecular biology. The next step is heat shock, which is used to sweep the plasmid DNA into the cells. So we start off cold, we plunge them into warm temperature, and in that process, the plasmid DNA gets swept inside the cells through the adhesion zones. And then we plate them on media on plates with antibiotics. Remember, the plasmids carry antibiotic resistance genes. So if we plate them on a Petri dish containing media with ampicillin or tetracycline, only the bacteria that have taken up the plasmid will be able to grow. After they've grown, we can then, on the Petri dish, we can then use the isolated colonies, grow up a lot of them, and purify a large quantity of plasmid DNA containing the DNA of interest, and then we can use that to sequence, or we can use it to make proteins, or as a probe for fluorescence in situ hybridization, or all kinds of things. Horizontal gene transfer is a very common occurrence. Here's a very interesting article all about it in Nature. Vertical gene transfer is the transfer of genes from mother to daughter cell or from parents to offspring. Horizontal gene transfer is the transfer of genes from one organism to another organism without being offspring of that organism. It can occur between organisms of the same species or different species. For example, a sizable fraction of bacterial genes are derived from horizontal gene transfer. Roughly 17% of E. coli and S. typhimurium genes are shared as a result of horizontal gene transfer during the past 100 million years. Another problem that we are being confronted with in today's world is antibiotic resistance that is acquired as a consequence of horizontal gene transfer. Bacterial resistance to antibiotics is a serious problem worldwide. In many countries, nearly 50% of strep pneumoniae strains are resistant to penicillin. So horizontal gene transfer between species and between different strains of bacteria is a very, very serious occurrence. In summary, bacterial phenotypes can be altered by changing their genotypes through the acquisition of DNA from other cells by conjugation, transduction, specialized transduction, and transformation. Generalized transduction occurs when a virus particle packages a random piece of host bacterial DNA into its capsid by mistake and then carries that DNA to a new bacteria cell. Specialized transduction occurs when a virus genome that has integrated into a bacterial genome imprecisely excises and takes a few adjacent genes with it. It then infects a new cell and injects the viral plus bacterial DNA into the new cell. Non-viral DNA transduced by viruses can integrate into the recipient genome via homologous recombination. Transformation occurs when bacterial cells take up DNA directly from the environment. This can happen randomly and is the mechanism of horizontal gene transfer. Transformation is also a technique used in the laboratory as a fundamental step in the molecular cloning of genes using plasmid DNA.